Hey everyone, quick biochemistry basics here. Let's talk about HPLC. HPLC is also known as high performance liquid chromatography or high pressure liquid chromatography. HPLC is usually a modified column chromatography. In column chromatography, a column is packed with an absorbent material like silica and the mobile phase is passed down the column because of gravity. But in HPLC, a high pressure pump is attached with the column. The high pressure pump can generate a high pressure up to 40 megapascals. The column is filled with an absorbent material which has a very small particle size. The small particle size gives a very large surface area for the sample molecules to interact. As a result, the efficiency of separation increases, giving high resolution. Let's see the components of HPLC. The column is made up of stainless steel which can withstand a very high pressure up to 50 megapascals. The length of the column can vary from 5 to 25 centimeters and have an internal diameter of 4.5 millimeter. The flow rate of mobile phase through the column is usually 1 to 3 ml per minute. The stationary phase. As discussed earlier, the stationary phase is made up of an adsorbent material that has a very small particle size. The particle size is kept uniform to obtain a better performance. Usually chemically modified silica, divinyl benzene, etc. are commonly used as a stationary phase. The mobile phase. The mixture of different solvents can be used as a mobile phase. The solvent used depends on the type of sample molecules which are to be separated. The mobile phase solvent can be polar or non-polar. The mobile phase is usually kept in the solvent reservoir. The solvent reservoir is attached with a pump which pumps the mobile phase in the column with a high pressure. Just before HPLC column, there is an injector which allows introduction of the sample in the column. The detector. In order to detect the eluate that is sample coming out of the column, the HPLC column is attached with a detector. Different types of detectors such as UV detector, IR detector, fluorescence detector, refractive index detector, mass spectrometer, electrochemical detectors, etc. can be used. The detectors are connected with a computer which collects the information. Now let's see the working of HPLC. As the sample molecules get separated, they are detected by the detector and a peak is obtained on the computer. This peak is plotted with respect to the retention time. To identify the components, we need to have standards. Let's understand this with an example. Let's say we run glucose as our sample and the peak of glucose is obtained at 5 minutes. Next, we run sucrose as our sample and the peak of sucrose is obtained at 8 minutes. Now we analyze the unknown sample for the detection of sugars present in it. Many peaks are obtained in the chromatogram. The peak obtained at 5 minutes indicates the presence of glucose. However, there is no peak at 8 minutes which indicates that sucrose was absent in our sample. The remaining 2 peaks are still unknown as we don't have any standards to compare their retention time. This is how the detection of sample is done in HPLC, and for the detection we must have the standards. Using HPLC, it is also possible to find the concentration of sample, but before doing so, we need to have standard curve. Let's say we take different concentration of glucose and run through HPLC. The peaks of glucose are obtained at 5 minutes, and with an increase in the concentration of glucose, the area under the curve increases. Once the standard information about the area under the curve and the concentration of glucose is obtained, just by measuring the area of glucose peak in the unknown sample, the concentration of the glucose can be estimated. Now let's see different types of HPLC. Normal phase HPLC. In this type of HPLC, the stationary phase is polar while the mobile phase is non-polar. Usually, silica is preferred as a stationary phase as silica has SiOH group, which gives its polar nature. During normal phase HPLC, polar molecules are retained on the stationary phase while the non-polar molecules move fast down the column with the mobile phase. Reverse phase HPLC. In this method, the stationary phase is non-polar while the mobile phase is polar. The non-polar molecules are retained on the stationary phase while the polar molecules move fast down the column. Size exclusion HPLC. This method is based on size exclusion chromatography or gel permeation chromatography. In this method, the stationary phase particles are porous. As a result, small molecules get inside the pores and takes a long time to move, while the large molecules easily pass down the column. Hence, separation occurs based on the molecular size. Ion exchange HPLC. In this method, the stationary phase has an ionic charge. If the molecules of interest has a positive charge, then a stationary phase is kept negative. And if the molecule of interest has a negative charge, then the stationary phase is kept positive. Hence, separation occurs based on the molecular charge. Here one quick biochemistry basics here. Let's talk about gas chromatography. Gas chromatography is a chromatography technique used for the separation of volatile compounds. Volatile compounds are the compounds that gets easily vaporized at room temperature. Let's see the components of gas chromatography. The column. The column used in gas chromatography is very long and arranged in a coil. The column used in gas chromatography are of two types. Number 1, pack column, and number 2, capillary column. Pack column. Pack column can be made up of glass or stainless steel. The length of this column can vary from 1 to 3 meters and has an internal diameter of 2 to 4 millimeters. The capillary column. The capillary column is made up of fused quartz. These columns are very long and have length from 10 to 100 meters and have an internal diameter of 0.1 to 1 millimeter. The column is placed in a chamber so that a uniform temperature can be maintained. The stationary phase. The stationary phase is packed in the inner wall of the column. The stationary phase is made up of silicon grease or wax which can withstand a high temperature. The mobile phase. The mobile phase use is usually an inert gas like helium or unreactive gas such as nitrogen. The mobile phase gas is kept in a cylinder which is connected with the column via molecular sieve. The molecular sieve separates the unwanted hydrocarbons, oxygen and water vapor that may interfere with the test sample during analysis. At the end of the column, there is a detector which detects the sample. Let's see the working of gas chromatography. The sample which is to be separated is mixed with an appropriate volatile solvent such as heptane, acetone or methanol. Just before the column, there is a septum that allows injection of the sample. The temperature of the injection region is kept 20 to 50 degrees Celsius high as compared to the column. This allows rapid volatilization of the sample. Once the sample is volatilized, it passes down the column where the separation occurs. During analysis, the temperature of the column is kept between 150 to 300 degrees Celsius. Separation occurs based on the interaction of molecules between the mobile phase and the stationary phase. The less volatile molecules interact more with the stationary phase and move slowly, while the more volatile molecules interact more with the mobile phase and moves fast down the column. Once the separation is completed, the detection is done with a detector attached at the end of the column. One of the most common detectors used with gas chromatography is flame ionization detector, or FID. The flame ionization detector has three inlets, one for the carrier gas which comes from the column and the other two for hydrogen and oxygen. The igniter ignites hydrogen and oxygen to produce a flame. When the sample molecules reaches the flame, they get ionized and the electrons are released. Across the flame there are two electrodes, each with a positive and a negative charge. The electrodes detect the electrons generated by the ionization of the sample. The electrons are detected in the form of current, which is amplified and detected by the computer. When the sample is detected, the computer gives a peak with respect to the retention time of the sample. The area under the peak gives information about the concentration of the sample. If the concentration is less, the area under the peak will be less, and if the concentration is more, the area under the peak will be more. Now, for the detection of unknown samples, we need to have standards. Let's understand this with an example. Suppose we run methanol as our sample and the peak of methanol is obtained at 5 minutes. Next, we run acetone as our sample and the peak of acetone is obtained at 3 minutes. Next, we run unknown sample and we get this chromatogram. Now, using information from the standards, we can say methanol is present in the unknown sample, whereas acetone is absent. The remaining peak is still unknown as we do not have any standards to compare its retention time. Here, one quick biochemistry basics here. Let's talk about paper chromatography. Paper chromatography is a common technique widely used for the separation of chlorophyll pigments, amino acids, DNA bases, etc. In paper chromatography, paper acts as a solid supporting material. The water molecules present in the pores of the paper acts as a stationary phase, while the mixture of different solvents such as ethanol, ether, etc. can be used as a mobile phase. The sample is applied on the paper using a capillary. Once the sample is applied, the chromatography can be carried out in different modes. The mode of operation of paper chromatography can be ascending mode, descending mode, radial mode, and a two-dimensional mode. In the ascending mode, the mobile phase solvent is kept at the bottom of the chamber. A small portion of paper is immersed in the mobile phase. The solvent in the mobile phase moves upwards due to the capillary reaction. During this process, the sample molecules get separated. Separation occurs because of the difference in the solubility of molecules in the mobile phase and the stationary phase. In the descending mode, the mobile phase solvent is kept on the top of the chamber. The solvent in the mobile phase moves downwards and separates the sample molecules. In case of radial paper chromatography, a wick is attached at the center of a circular paper. This wick remains submerged in the mobile phase. The wick absorbs the mobile phase solvent by capillary action. As the mobile phase moves throughout the paper in a radial fashion, the separation of the sample occurs radially. In case of two-dimensional paper chromatography, the chromatography is first carried out in one dimension. Once the chromatography is over, the paper is now rotated by 90 degrees and the chromatography is again carried out. The two-dimensional paper chromatography is widely used for the separation of amino acids.
When running reactions in the organic chemistry lab, you want to have a way to monitor them. One of those methods is known as thin layer chromatography, or TLC for short. This works like all other kinds of chromatography in that you have a mobile phase and a stationary phase, and the compounds that you're trying to separate interact with these two phases. First, let's talk about the stationary phase. In TLC, you usually have a plate that's completely coated with silica gel. The silica gel is the stationary phase. There are other materials you could use, but silica gel is the most common. What you would do first is take a spotter and dip it into your reaction flask, then put a little spot here. You want to find out whether is there one compound in there or are there multiple compounds. Next, what you'll need is to prepare the mobile phase. This is done in something known as the developing chamber, shown here in white. First, what you would do is put in a tiny bit of your mobile phase, meaning it can be any solvent or any mixture of solvents, and you want to keep this to be a relatively small amount. If the level of this is too high, your spot will be completely submerged in this. You'll want to put in a piece of paper that will indicate whether or not this chamber is saturated. When setting this up, you want to check that this piece of paper is wet. This lets you know that the vapors from the mobile phase are completely everywhere in the developing chamber. Next, what you do is you take your TLC plate and put it inside here. Finally, you would close the top. You want to do this because if you didn't and you left the top open, organic solvents are very volatile, meaning that they readily evaporate from liquid into gas. Next, what would you observe? Well, let's think about what could happen. You have your pink mobile phase, and what will happen next is that it'll travel up the TLC plate through capillary action. Capillary action is just a term for when you have some kind of solid thing, like silica gel, that sucks up a liquid. And when you see that it's gotten pretty close to the edge, say around here, you'll want to pull this out of the developing chamber. Make sure to use a pencil to mark where your mobile phase got up to. How come we can't see anything yet? That's because usually you need something like a UV lamp shining on this so that you can visualize something. So let's say we take our UV lamp and shine it on here. Compounds that are aromatic will usually show up and fluoresce. And let's say that we had these two dots. So what does that tell us? That tells us that whatever was in our reaction flask was a two-component mixture and that there's at least two compounds in there. However, you would have seen more dots if there were 3 compounds or 4 compounds or even more. TLC plates can get really messy when you're doing research. But what can we tell about these 2 compounds? So TLC is a pretty qualitative method. It'll tell us whether things are more polar or less polar. So what we have to keep in mind is that the stationary phase, or the silica gel is very, very polar. So you can see this one didn't move too far. Means it must have been really attracted to the silica gel. But this one moved a lot more. So this is less polar and more attracted to the mobile phase. Okay, so we still don't really know what compounds these are exactly. But let's say that you knew that inside the flask you had naphthalene and benzoic acid. How can we tell which spots these correspond to? Well, you'd want to look at them and figure out which one is more polar and which one is less polar. As you can see, naphthalene is just made out of carbons and hydrogens, so it's pretty nonpolar. So we can indicate that this corresponds with this, and because of the carboxyl group in the benzoic acid, this group right here, you can tell that it's a pretty polar molecule. So this would correspond to this. And that's how you do a TLC in a lab. Again, let's review. You first have your plate that you put a little spot on. You put it into the developing chamber, which contains the mobile phase. Wait a little bit for the mobile phase to travel upwards. Pull it out of the chamber, and then use a UV lamp to see what spots are there, and then try to compare those two spots in terms of polarity.